From the Heart.org Medscape Cardiology, this is the Bob Harrington Show. Dr. Robert Harrington is the Arthur L. Bloomfield Professor and Chair of Medicine at Stanford University.
Hi, my name is Bob Harrington from Stanford University. Welcome to our podcast on Medscape Cardiology and the Heart.org. You know, over the course of the many years that I've been doing this, I've had the privilege of getting to interview a series of friends and colleagues who double as both physicians and authors. And it's been really fun to talk with them about the book or the essays or the ongoing writing journey that they're on, as well as trying to understand how do they put that in the context of being a physician? How does that add to the richness of their professional life, for example? And it's really been fun. And I can tell you from the feedback we get on the podcast that they're some of the favorite interviews that I do. So today I'm really privileged to not just interview a physician author, but interview somebody who's a friend, a colleague here at Stanford University, and really talk about his new book. The book, I have it right here, is The Genome Odyssey: Medical Mysteries and the Incredible Quest to Solve Them. I had the privilege of reading this book over the last couple of days, and so that I was prepared to talk to my colleague today. He's a practicing cardiologist. He is an active cardiovascular researcher who runs a large laboratory that I suspect will get into some of the topics. He's the founding director of the Stanford Inherited Cardiovascular Disease Clinic. He is a site and a leader nationally in the NIH's Undiagnosed Disease Network. And he, like many other Stanford faculty members, is the founder and entrepreneur of behind several local biotechnology companies. So Ewan, it's a privilege to have you here. Thanks for joining us on Medscape Cardiology, and I look forward to the conversation.
Oh, it's my pleasure to be here, Bob. Thank you so much for having me. I love the podcast, so it's just my privilege to be on it.
Well, now that I just reminded myself of the Scottish accent, I left out the most important part of your bio, that you have an ongoing research interest in single malt Scotch whiskies. So perhaps we'll come back to that.
Yeah, that may be the most important of my research interests.
I was pleased to see that it made the jacket cover of the book.
Yeah. It's usually the thing that people remember the most.
So, as I said, I had the privilege of reading your book this weekend, and I have to say, as a fellow nerd, and you describe yourself in some ways as a nerd throughout the book, I loved the science. I loved the history of science, but I also really appreciated what a great story you told and the language that you used. And I don't know, is there something in the Scottish personality that makes one a natural writer?
Well, that's very kind of you to say, and I'm not sure about something in our background or heritage. I know that certainly the power of narrative is so strong, and I think it really pervades all of medicine. We're constantly telling each other stories about patients. Every morning reports a story. Every time you talk to any other colleague about a patient, it is a story. And so I do kind of see the natural narrative in medicine, and I think that's what people cling on to. And I was hopeful to write a book that included science, as you mentioned, but that was really framed around real people, real patient stories. And as I passed it around my various family members before it ever got to be read by anyone else, it was often those human stories were what they commented on and that they really enjoyed the most.
Oh, and there are some great ones, and I'm going to pick out a few of my favorites and ask you about them, because it is the power of narrative. And our mutual friend Abraham Verghese talks about this. He's been on this podcast and is one of the country's great writers who really does believe, as you've noted, in the power of narrative as a way of teaching medicine. So I'm glad that you've made reference to that. Ewan, let's not assume anything. So let's start with the basics.
Yep.
The Genome Odyssey. Some of our audience will be hardcore scientists who say, well, I can tell you about the genome, but not everybody will. So why don't you level the playing field and tell us, what is the genome and why does it fascinate you?
Yeah, well, the genome is the blueprint within almost every cell in our body. Red blood cells don't have nuclei, so they don't have the genome. But it is our own individual, completely unique code And so people are probably aware that identical twins share basically a genetic code, but it's not actually even among identical twins identical. So everybody has their own unique code. It connects us at some level to every living organism on Earth, connects us to the history of the human race. And it is in its essence 6 billion letters. And each one of them is an A, T, a C, or a G. And I think that part of me, you mentioned my nerd-like tendency, but I think part of what attracted me to the genome is that it is a computable thing. It is 6 billion bases of data. And I was drawn early on as technology allowed us to start to sequence when we spell out the letters of the genome at a speed and a rate that we just couldn't do before. That I was just watching technology advance just in at a speed beyond anything I'd witnessed in certainly my history of science or medicine, and it just seemed so exciting. I wanted to jump on and see what I could contribute. So, in its essence, it contains so much. There isn't a disease that doesn't have a genetic component. There isn't an aspect of your human body that doesn't have a genetic component. We also have to always balance with the environment that we're in, but there's a genetic component to so much of what we are and what we do in medicine that it just drew me in and fascinated me for years now.
Yeah, and let me for the people who have not yet read the book but who are thinking of it, that to me was one of the things that captured me, is that your fascination with it comes across. And I loved the dropping of the occasional fact, like if you were to unravel that 6 billion base pairs, it forms a length that would go to the moon and back multiple times. And so those sorts of things, I think, capture the reader's attention. And again, as you say, for somebody like you, interested in science from a young age, with parents who are in the field, really does come across.
Yeah. No, I think that much of, in fact, my interest in science was actually driven by reading popular science books. I mean, I had great teachers at school, but in reality, it was actually outside of class that I really got a fascination with science. And my biology teacher, who, just to be clear, thought I was a total buffoon and would amount to nothing, and told my parents that at one parents' evening, did me another favor, which was that he gave me a copy of Richard Dawkins' The Selfish Gene, which was just one of the most impactful books, including the origin of the word meme, actually, for any of our younger listeners. Yes, the origin of that was in the last chapter of that book. Now it means something a little different from what he coined. But what I got from his book early on was just his fascination with the natural world and his fascination with science, and his explanatory style just drew me in. And I hoped, one day it might be possible to try and ape that a little bit and potentially write my own book one day.
Well, I was pleased that you told the story of your biology teacher who thought you might amount to very little. And it's nice to see that you've proven them wrong, which is a good lesson for many of our listeners out there who to continue to follow their love, follow their passion, because it usually does persevere. And that's a good lesson that's in the book. So before I go through the 4 sections of the book, which again, I found really helpful, but I'm amazed you, in each one of those sections could have been a book in and of themselves.
Yeah, I mean, I think when I started writing, I honestly didn't know if I was writing a book. I knew I had a sense of stories that I wanted to tell and to spread the word, and I hoped that others who would read patient stories would gain something from them. But I had and I knew I could write scientific papers. I've written plenty of those over the years, but this is a whole different style of writing. And so I thought probably at some point I might try and write a book, but I had that, pegged at some distant moment, in a sabbatical in 10 or 20 years, or, retirement or something like that. And there was actually, there's this moment in time. In fact, we were lucky enough to spend some time with the Science and Technology Policy Office of the Obama administration, as you know well, Bob. And I had taken a couple of my students to this event in Pittsburgh, at which Atul Gawande and the president were present. And Atul Gawande was interviewing President Obama on stage. And Obama, I think we all realized towards the end of his presidency in particular, is also a nerd. He just loved science and technology. And this came across so much in this particular event. And I think it was basically in the audience of that event that I realized these two very inspiring writers and humans talking to each other. I just felt at that moment that I shouldn't wait, and I should actually try and write. And so on the way home on the plane, I basically just started writing. And I wrote, with no sense of where it was going to go. But as you mentioned, each of the individual sections could have turned into a book. It was for great editing that it managed to sort of keep it in place and try and provide some structure to it. And I've learned a huge amount from the editors.
So let's talk about that, the process of writing, and then I'll get into some of my specific questions about the book. You say you just started writing, and that usually is because somebody has a story that they want to tell. But it fits so nicely with the 4 sections. Did you come up with that, or did your editors say, look, this really falls nicely into 4 discrete sections? What was that moment?
Yeah, no, it's a really good point and great observation, because I had, as I mentioned, I just started like getting some stories down, and I thought I was hopeful that these were ones that a reader would find interesting. But yeah, huge credit to my agent and to my editor at the publishing house, who said, we see a lot of potential here, but it's a little unformed. And if someone's going to read a book, they want to feel that there's some sense of narrative arc from beginning to end. And so in some ways, the beginning part was easy, in the sense that the early genomes were going to come first, because you can't, the rest of the book depends on that. And in fact, the first story with my colleague Steve Quake also contained elements of around his heart disease specifically. And so that provided a nice entryway into explaining for the general reader what an ECG was and what an echocardiogram was, and concepts that would come to be revisited throughout the rest of the book. So that part I think I could have done almost by myself, but what really helped was having these experts and just consummate professionals in the publishing world who really just understood books and understood the reader's perspective to say, well, obviously, if we start with the early stuff, the last part should be facing towards the future. And how do we now organize these stories in the middle, which were a little bit of a large grab bag of things, and say, how do we organize them in a way that will make sense for the reader? And eventually we divided them into kind of disease detective stories, and then some specific inherited cardiovascular disease stories, all with the genome as a constant thread. But these different sections basically were suggested themselves in the end.
Well, it comes together beautifully. And let's go to something that you already mentioned, which is the Steve Quake story, which is, as you've pointed out, we've had the privilege to live through a lot of, we'll call it genomic history in a short period of time. Tell the Steve Quake story, because it's fascinating. Number one, that you wander into his office and he's looking at his genome. Right. Number two, that then you assemble really, and you called it, I think, an all-star team to do this. I think people might be amazed at how many people and the man hours, the woman hours it took to get this all done. So tell the story, because I think it is the entryway into the book, and so maybe it's the entryway into hearing about the book.
Yeah, no, absolutely, and it was a major turning point for me, for sure, and I think a little bit for Steve as well. Steve is a colleague and now friend, although at the time I did not know him. He was already a renowned bioengineer. He was chair of the bioengineering department at Stanford. And we were planning, I think, a seminar on genetics, because our new chair was coming, Mike Snyder was coming. And so I wandered over to his office. And I remember it well, first of all, because that building, as you know well, Bob, the Clark building at Stanford has a very gratuitously complicated numbering system. So finding anybody's office in that building, first of all, is an achievement. So I felt that I had achieved something when I got to his door and found him. But I always had this crystallized picture of him in his office, because it's one of those offices where all the journals from the last 10 years, which are all electronic now, but by then he had piles of journals all over the place. You could barely see him. And in the corner of his office, then he was there like pecking away at his keyboard, this like world famous scientist. And I came in to talk about this very sort of bland topic of like a seminar we were planning. And instead he had other ideas, and he said, hey, come on, look at this. And he points to me at his screen, and it's basically his genome on the screen. And I'm seeing A's, T's, G's, and C's, and he's pointing at gene names. And he ends up pointing at a gene I knew quite well. Now, there's 20,000 genes. He could have pointed at anyone, but he points at myosin binding protein C, which is one of the genes that causes hypertrophic cardiomyopathy. And he says, hey, look, I've got a variant in that. What do you think about that? And I was like, well, it could be nothing. It could be something. And I started just kind of asking him about his family history. And And suddenly he's telling me about a quite significant family history of heart disease, including his dad, who has VT, ventricular tachycardia, quite a lot of coronary disease, strokes. And then I asked him this sort of key question in relation to inherited heart disease, which is, did anyone in your family ever die suddenly? And he said, well, it's funny you should say that, but my cousin's son died suddenly, age 19. Black belt in karate, and just didn't wake up one morning. And so suddenly the whole thing changed. I mean, I was having this incredible moment in science where only the fifth person in the world to ever have his genome sequence was sitting in front of me, pecking at his keyboard, and my jaw was on the floor, like looking at the genome. But all of a sudden, we switched back into doctor mode, because now I'm faced with someone with a very significant family history of heart disease, and even including early sudden death of a teenager. And he's literally showing me genes I know and variants in those genes I know cause a disease that is associated with sudden death. And so it switched in a moment, and I was suddenly essentially inviting him to our clinic to become a patient. And he's saying, well, that's funny you should say that. My family's been telling me I should go see a cardiologist. And I'm like, well, you have one now. But then the dawning realization happened as I left the meeting and walked back to the cardiovascular building, where I realized that a patient was about to walk into a clinic with their genome, essentially on a hard drive, like walking into a clinic with the whole genome. He's only like the fifth person in the world to ever have his genome sequenced. And the realization was he was walking to our clinic. we were that team. And that was daunting and terrifying. Because, as you know well, we're used to looking at lipid panels, with 5 numbers on them, or a CBC, or Chem7,
and looking at the family history with little circles and squares, and trying to identify relationships. But now, yes, you had relationships, but you also had the source code.
Yeah, just an incredible moment. And just, I mean, for context, remember the Human Genome Program was 10 years between 1990 and about 2001. And it was $3 billion of funding. It was 10 countries, 10 years to do one. And it wasn't even one, as I described in the book. It's actually half a genome, but more on that another time. But, I think that, and this was only like 7 or 8 years after that. And suddenly we'd only moved on. There were like 4 or 5 more people who'd been sequenced. But it equally looked like the beginning of something really exciting. And I don't think I could have guessed what was going to happen in the following 10 years. But what seemed really clear was that genomes were going to become more available and more affordable. And given, as we talked at the beginning, that every disease has a genetic component, it just seemed inevitable that we needed to be ready to be prepared for that world, and that we were singularly not ready to prepare for a world. If we're used to looking at 5 numbers and suddenly you have 6 billion letters and you're trying to parse that in the context of a short clinical visit, how would you even do that? And so that was the question that I was facing. And of course, being an amazing place like Stanford, just so lucky to be here in this environment, I thought quite quickly of some colleagues who could help. And I called them up basically and said, I have this crazy thing, Steve. And they're like, yeah, we've heard of him. He has his genome, and there's a family history, and there's more than that. And we need to do a workup, and we've got that going, but we need help to parse a whole genome in the context of a single patient zero, we called them at the time. And people just came. It was amazing. Another amazing thing. They didn't show up themselves. People like Atul Butte, who was at Stanford at the time, now at UCSF, folks like Russ Altman, basically brought not just themselves, but they brought like a team of 15 people to the meeting. And suddenly there was like, you crammed into this little conference room as like 30 people all trying to work on a single genome of a single patient who was also in the room because he was part of the team. It was just kind of.
Well, that was the amazing part of the story for me as a researcher, that it was the moment where you said there's something here, and everybody just rolled up their sleeves, it sounded like, and devoted their expertise. You want to talk about doing unfunded research. This was the ultimate, that you got a group of all-stars to give their time, their energy, their expertise. It's extraordinary. And but let me move on to some of the other topics, because the Steve Quake story is a great intro. And then in part 2, you start to get into what you call the disease detectives. And people, like, after you become a genome celebrity with the Steve Quake story, people start seeking you out. And you start learning all sorts of things when they would ask you questions about themselves, their family, their children. Does this happen any place other than Silicon Valley? And What's the role that Silicon Valley you think played in this evolution over the next 10 years of the work that you were going to do?
Yeah, it's a great question. And I think that certainly in those early days, Silicon Valley was awash with people who really wanted to be on the cutting edge and had the resources to take them to the cutting edge. And so we really were getting calls all the time from people who had realized, sequencing was going to be a thing. Maybe they had a family history. Maybe they were just more interested in preventive care and wondering what was hiding in their genome. And so, yeah, they would call us, and many times they were CEOs of large companies that many of us would have heard of, and so they were looking for some. In some cases, it was the next big thing, clearly, and so they just wanted to be part of that. And then in other cases, there was very specific questions related to family history or very specific questions related to future preventive healthcare. And so I think that was fairly unique. We heard some stories from New York at the time of some similarly high level folks who were looking for that kind of interpretation. And so there were a handful of groups in the country at that point that were putting together pipelines that could potentially interpret a human genome in a way that would be helpful. But I think there was a certain uniqueness about the desire to kind of change the world in a slightly unrealistic way that is part of Silicon Valley, the highly collaborative part, and then the willingness of people to kind of sit there on the cutting edge and really look to see what does the world look like. I say at one point in the book that a sort of futuristic tendency is a powerful driver of a creative ideation. And I think it sounds like a lot of big words, but what I mean by that is if you're someone who can imagine a different future, then you're almost forced to imagine how do you get from here to there. And of course, those are different skill sets in a way. Imagining the future is kind of fun, and how you get from here to there is a little bit more, nuts and bolts of how you get there. But I do think there was a uniqueness about that time and place, and the also unique interactions, I think, between Stanford and the Silicon Valley that surrounds it.
Yeah, and that comes across. I mean, as you talk about, for example, the founding of one of your companies, Personalis.
Yeah.
I mean, there are some great stories for the readers who are interested in how the biotechnology sort of world emerges. So some good stories there. Let's get into section 3, because I will say that I thought section 3 was it might have been my favorite section. And maybe I say that because, like you, I'm a clinician.
Yeah.
But these are really the essence of narrative. And the two stories I want you to think about are Ricky and Lalani. And now, in full disclosure, Lalani, I know through the American Heart Association, where she has been a powerful voice for patient advocacy. The stories are amazing for different reasons. One of the things I really enjoyed about the Lalani story was how you weaved into it the whole history, again, as an AHA person, to hear about the CPR and the beginnings of defibrillation, and this young woman who is an NYU student and is saved by defibrillation. I mean, and then that ties you into hypertrophic cardiomyopathy and Jim Sputich and biochemistry. I mean, how you weaved all of this in a way that we could read it. It's an extraordinary story. So just say a little bit about Ricky. I left that story for you, and then maybe comment a little bit on Lalani.
Well, yeah, but you're so kind, Bob. But I think that telling these stories is so much my privilege. And when I think about what our patients go through, and in this case, these two remarkable young human beings and what they've lived through from such an early age, it's just awe-inspiring, honestly. And I think that being able to try to tell those stories, and as you say, weave in the aspects that we find so fascinating as doctors and scientists that have led to the major advancements that have allowed us to bring life-saving therapy to these individuals was really just too enticing to pass up, and I hope I managed to do it justice. You were asking about Ricky. I think a very unusual individual in that he had a condition that would have appeared to be something called carne complex, and there's many aspects to that, but neuroendocrine tumors and myxomas, cardiac tumors, are a major part of it. And so he first came to the attention of one of our colleagues, Dan Bernstein, in pediatrics when he was younger, just in his early years, 5 or 6 years old, because he had a very large tumor in his right ventricle that was bobbing in and out through his tricuspid valve. And basically a fill-in GP, they'd gone to see a different GP because he was having some palpitations. And that individual did a clinical exam and heard this plopping, bizarre murmur and said, I don't know what that is, but you need to go see someone. And so
the
he ended up,
classic plop that we're told
yeah.
about myxomas, right?
And this was a locum GP that was filling in. So that's where his story began. And he had an initial heart surgery. He had a testicular tumor that was removed. He had pituitary tumor that was removed. He had another heart surgery. He had another heart surgery. And then when we met him, I remember, and I think that we start part of the story with this, is that we were waiting for our next clinic patient. And, sometimes you're well informed and you've looked in advance, and sometimes you're just like, who's coming next? And I swiveled around and looked at the screen, and this long axis view of his left ventricle was there, and there were two massive golf balls bobbing around in his left ventricle. And it's one of those things where you do a double take and you're like, that can't be real. And suddenly everybody in the room swiveled over, and both in the outflow tract of his left ventricle and in what looked like the subvalvular apparatus of his mitral valve, there were two large myxomas. And that's where our story with him began. And he's a very, just an incredible young man. The story goes through a number of twists and turns, including him being on the transplant list, including him deciding that he wanted to take himself off the transplant list, including at one point, one of his close relatives dying in an accident and looking to donate their heart to him, and a number of other just remarkable things. But it leads up to a moment at the end that I know we chatted a little bit about earlier, where we had to make a decision. Was he going to be a heart transplant candidate, or was there some kind of heroic surgery that could be done?
Yeah, and that's the story. I don't want to go through the details. I'll let the reader have the pleasure of reading it. But I was impressed and not surprised by your powers of observation when you describe our mutual friend and colleague, Joe Wu. And I could hear his voice the way you wrote it, which is, first off, Joe always wears a tie.
Yeah.
And you noted that he had, pulled on his tie, which Joe does.
He does.
Before he says something. And, and in classic Joe Wu fashion, he just says, let's get this done. And I mean, how many times do you call Joe and you say, Joe, I got this difficult patient? Well, let's get it done. Let's, send him over. And I thought it was a great story, and for a great surgeon. But I won't spoil the story. You should go ahead and read it.
Yeah, we'll let the readers go. I have another story just about Joe, just since you mentioned it. It's not in the book, but I loved it just because it reflects exactly the same issue. I mean, we had it's the only time I've been on the helipad actually at Stanford, and I was on for the CCU, and we had this patient who basically had an anesthesiologist with them being transferred in the helicopter, and they'd had essentially a series of tension pneumothoraces in the helicopter, basically. And this an anesthesiologist had to basically put in cannulas 2 or 3 times to try and stabilize the patient. They had an impella in the axillary artery. And was called up to the helipad. I was like, why do they want me on the helipad? You probably need someone else. I mean, I am the CCU attending, but so it became clear the plane landed this tiny little space inside this helipad, and I got the immediate story from the anesthesiologist, and we're like, clearly this patient needs an operation. So we basically called Joe with insight from inside of the helicopter on the roof. It was still slowing down the rotors. And this is a man who gets things done. And he had basically described the story. He said, give me 2 minutes. I'm like, okay, well, that was a long 2 minutes. And he just called back, and all I heard at the end of the phone was OR8. Done. And we just put the patient straight from the helipad into the operating room and saved the patient in the end due to his team's amazing work. And so I think that sometimes you need a surgeon to get it done. He's often the guy to help.
What I also appreciated is that as you've said, that you're telling all of this elegant science, and then at the end of the day, what you really need is the surgeon to just get this done. And and it's remarkable. As we get into the last section, Ewan, in our last couple of minutes, I'm going to let you pick. Do you want to tell the story of elite athletes, or do you want to tell the story of PCSK9?
Well, I'm thinking that your listeners might have heard quite a lot about PCSK9, although I love that story too. But I guess that the elite athlete was such an unusual one So
Yeah,
I don't
go
know.
ahead.
Shall we tell that one? I mean, this is a story that has just fascinated me, but it really is at the fundamental, and I deal with it in the part of the book where we talk about how the genome is used for drug discovery, and that's part of the PCSK9 story. But this was another Olympic athlete who'd won more medals than anyone else in cross country skiing. He came from Lapland, and he basically grew up doing cross country skiing to get to school in the morning and then in the evening. Remarkable, he won all these medals. Multiple gold medals, was like 15 seconds ahead of the nearest competitor. But this cloud hung over him, suggesting that he might be blood doping, because every time his blood was taken, he had this really high hematocrit. And that's the only way you can tell, because if people know, blood doping is you remove blood from yourself, you store it, and you give it back. And those red blood cells obviously carry more oxygen and are performance enhancing. Similar idea behind EPO supplementation or even altitude training. And basically he had just this really high hematocrit, just beyond anything that anyone could consider. So this whole cloud, and at one point he was actually banned. But what it turned out to be the case was that he actually, when several other members of his family also were found to be these remarkable athletes, that he actually had a genetic mutation in his EPO receptor. He was actually kind of a genetic superhuman. And what it meant was that his EPO system was basically like pedal to the metal, basically. The accelerator was down the whole time, and his bone marrow was just making red blood cells like constantly. And it turned out that he had this kind of superhuman mutation. And in the end, when this was laid out and his whole family tree was there was a paper in PNAS, and essentially he was exonerated as science proved. What he said all along, that he'd never done any blood doping. It's just a remarkable story. Iro Mantaranta was his name.
It is a great story, and I thought that there's a lot of great stories at the end. I mean, gene therapy, CRISPR, just great stories that people should go and read. But I did particularly like the inherited endurance, which is just a great story to tell. Well, you and I could talk about this all afternoon. It's a terrific book. My guest on Medscape Cardiology is Ewan Ashley from Stanford University, and the book is The Genome Odyssey. So Ewan, thank you for talking with me. I know you've been busy with this, but we appreciate it. And like I said, I can really endorse reading it. I had a wonderful time reading it this weekend. So congratulations.
Thanks so much, and thanks for having me on.